Although it is clear that TLRs play a key role by recognizing pathogen-associated molecular patterns and triggering an efficient immune response, this particular study presents evidence suggesting that TLR3 may be contributing to influenza A virus pathogenesis, since mice lacking the receptors displayed a clear advantage over the wild-type population. This was seen in the extended survival rate by mice deficient in TLR3 over controls. Furthermore, TLR3 mutants also experience significantly reduced levels of inflammatory mediators, as well as reduced leukocytes to the bronchoalveolar spaces. Now, the authors state that similar previous research has not been successful at establishing individual roles for given cytokines in influenza-induced inflammation, since there is redundancy between cytokines. Therefore, inhibiting one type seems to have partial effects on the overall result, as other cytokines might be involved in the same signaling pathway. Nonetheless, the present study compiles substantial evidence related to the involvement of TLR3 in triggering the inflammatory response to influenza virus, since in the absence of the receptor, significantly lower levels of interleukin-6, interleukin-12, and regulated upon activation T cells were reported. The investigators did find that expression of other mediators was increased in the absence of TLR3, but agreed that to hypothesize on the mechanism of action for this differential pathway would be too complicated at the moment, in part due to the already mentioned cytokine redundancy. It seems clear that in the absence of TLR3, there is a reduction in lung injury due to a reduction of inflammatory mediators and CD8 cells, which results in increased survival. This observation has also been obtained in previous studies where mice had been treated with immunosuppressant drugs before infection with influenza A virus, and they also experienced less pulmonary pathology. However, it is noteworthy that the mortality rate experienced by the TLR3 mutant, although lower than in controls, it was still high with high viral doses of influenza. Therefore, we can see that TLR3 plays a part in a negative immune response to influenza virus, but it varies with varying viral doses. To sum up, it was found that reduced TLR3 mediated inflammatory infiltrate ends in reduced clinical effects of influenza A virus-induced pneumonia. But to only focus on downregulating the receptor would not be appropriate, since in those cases the viral load was higher. Thus, the authors suggest that vaccination should remain the main method for influenza prophylaxis, along with viral replication inhibitors, but administering modulators of TLR3 in high-risk populations may serve as an effective addition to the treatment.